They raised money, built roadmaps, and promised to bring back a bird last seen in 1932. After all the buzz, the embryo count is still 0. That is the Heath Hen De-Extinction story in one line. Millions in plans, 0 embryos. A comeback that looks more and more like the project that died twice. Picture this, a lost bird from Martha's Vineyard, last seen booming on the lek in the early 1900s, suddenly put back on the table as a model comeback for North America. The heath hen, a stocky prairie grouse once so common it was poor people's food, is now a ghost remembered in field guides and in a single statue nicknamed Booming Ben. Scientists and funders looked at his story and said this could be the gateway bird for de-extinction, the proof that modern genetics can turn an ending into a second act. On slides and in press releases, it sounded unstoppable. In reality, the timeline stretched, the updates thinned out, and work quietly stalled out well before a single edited embryo reached a lab dish. Receipt: milestones on paper, no embryos in the lab. It was pitched as the model project. The model showed up, the actual bird did not. Receipts first, verdict later. By the end of this episode, I want you to have one clear takeaway you can drop into any argument about just bringing species back. How a project can look huge on decks and headlines, yet still deliver exactly 0 chicks. Wild ones. This is not a hit piece, it is a case file. We are going to walk through what the plan actually was, what got done, what stalled, and whether it deserves a second life or a firm, not like this. Then we will put a simple label on it, the same as every other episode in the series. Greenlight or Veto. Let's start with the sales pitch. On paper, the plan sounds neat. Step 1. Sequence as much heath hen DNA as you can from museum skins and old feathers. Then compare it to its closest living relatives, the greater prairie-chicken and other prairie grouse. The team wanted to know whether the heath hen was simply a coastal form of prairie-chicken or something more isolated that needed its own label. That meant checking how its DNA lined up against today's birds, not just how its plumage looked in old paintings and dusty photos. Step 2 in the pitch, build a draft genome good enough for editing. You do not need to resurrect every single letter perfectly. You need a clear working map, especially for the genes that shape the body, feathers, and immune system. That draft can then guide edits in living cells from prairie chickens or other close relatives. By the early 2020s, the heath hen sat on de-extinction project lists with draft genomes in hand and plans to improve them as sequencing tools got sharper. Step 3. Move from maps to bodies. The idea was to edit primordial germ cells, the early cells that later become eggs and sperm from a living prairie chicken or a similar galliform. The adult bird would look normal on the outside, but his ovaries or testes would quietly produce heath hen coated eggs or sperm. Pair 2 such birds, collect eggs, hatch chicks, and over generations breed toward a heath hen like bird that can lek, boom, and forage in coastal shrubs again. Germ cell editing had already been demonstrated in chickens and quail, so the pitch could say, we are not starting from 0, we are extending a lab trick into a wild relative. On a conference slide, this all compresses into 3 arrows: extinct DNA to genome, genome to edited germ cells to chicks. But each arrow hides years of work in ancient DNA handling, delicate cell culture, and bird reproductive biology. When you pull the pitch apart, it stops being a clean, we can bring the backstory, and turns into something more honest, conditional, slow, and dependent on a long list of technical and financial ifs. Claim tidy, lift heavy. So where did this polished roadmap get us in the real world? The early story is all momentum. Around the mid-2010s, de-extinction groups held talks on Martha's Vineyard, promoted the heath hen as a serious candidate, and pitched it as a gateway bird that could open the door for other extinct species. Private donors and foundations signed on. Articles in local and national outlets. talked about the bird that might boom again on the island, and Outreach framed it as a way for people to reconnect with a lost piece of North American ecology. Behind the scenes, scientists worked through the concept phase. They needed basic answers first. Genetically, was the heath hen distinct from mainland prairie chickens? And which living birds would make the best surrogate template? Population genetic work suggested the vineyard birds were more isolated than today's prairie grouse, which meant any proxy would need careful design, not just grabbing any farmyard chicken and calling it done. That kind of framing matters, because it sets expectations about whether you are rebuilding a lost population or designing a new hybrid that only wears the old name. On the lab side, progress was slower and more fragile than the headlines. Phase 1, sequencing and work to compare the genomics moved ahead. But when the project tried to push into deeper lab work, including avian germ cell culture with a company partner, it hit a wall that had nothing to do with the bird. Business reality. The partner company doing core avian genetics work was acquired, and the heath hen work was stopped as the lab's priorities changed. The genomes did not vanish, but the engine that was meant to turn ideas into eggs simply switched off. Today, the heath hen still appears in de-extinction roundups as a potential case, and its genome resources are described as templates for conservation genetics. But there are no embryos, no chicks, no captive flocks in hidden barns, and no date on the calendar when a return is expected. In other words, the story now lives as code, concept notes, and museum skins. On paper, it is not dead, but in practice, it is paused. Chapter verdict: active idea, stalled project. Now we need to talk about the birds that would actually carry this plan on their backs and in their bodies. Heath hens were galliforms, heavy-bodied, ground-dwelling birds in the same broad club as chickens, turkeys, and grouse. That is good news on one level, because farm birds in this group have been studied for decades. We know a lot about eggs, clutches, incubation temperatures, and artificial insemination, but the more you zoom in, the more rough edges you find between tidy theory and real surrogates' life. First, there is the basic reproductive math. Prairie chickens and related grouse have modest clutch sizes and fussy breeding behaviour. They gather on leks, males perform elaborate dances, and females pick mates, then nest on the ground in exposed habitat. The breeding plan expects calm, cooperative birds. The birds naturally have not read the breeding plan. Turning those wild birds into reliable factory surrogates for edited germ cells is not like flipping a switch on an industrial chicken farm. It likely means capturing and handling wild or semi-wild birds, or building captive flocks and managing them for years, just to get a handful of viable, edited offspring from each generation. Second, stress and welfare. Galliforms can be surprisingly fragile when it comes to stress, disease, and handling. Each step, from taking biopsies for germ cells, to keeping birds in controlled environments, to repeated egg collection, piles extra risk on individuals. You are asking each animal to live its own life while also acting as a piece of biological lab equipment. Every time a bird is restrained, injected, or moved, there is a chance of injury or death that never appears on the glossy timeline graphic in a funding deck. Then there is the cumulative risk of running this over many generations. To get anything close to a heath hen-like bird, you would not stop at one edited clutch. You would repeat the process, refine edits, cross birds, and select for traits, all while guarding against inbreeding and keeping disease out of your flocks. Every breeding season runs through the same stresses: capture, sampling or surgery, egg retrieval, and human-managed incubation. If you imagine a long production line, the weak points are not the machines, they are the living, breathing hosts. Until avian germline editing becomes much more efficient, Each step of this plan leans heavily on real birds with real limits. That does not make the idea unethical by default, but it means any Greenlight has to include a welfare budget, not just a dollar budget. If the likely outcome is a small number of proxy chickens at the cost of many stressed or lost surrogates, the math starts to look less like repair and more like a very expensive, very demanding experiment. Call here. Big welfare load, unclear payoff. Even if you could turn genomes into chicks in a clean line, those birds need somewhere to live. Historically, heath hens lived in scrubby coastal barrens from New Hampshire down to Virginia, but by the time the species collapsed, their last stronghold was Martha's Vineyard. That island is also where the modern de-extinction story was anchored. Talks with the local community, discussions of restoring shrubby heathland, and visions of tourists once again watching males boom at dawn on protected leks. In a brochure, it sounds poetic, like history coming full circle. Reality is less romantic. The original heath hen reserve on the island saw the species spike, then crash, thanks to a mix of wildfire, harsh winters, predators like goshawks, disease brought by domestic poultry, and plain bad luck. Those pressures have not all gone away. Modern Martha's Vineyard has more people, more infrastructure, and a very different fire and predator regime than it did a century ago. Before you release any proxy birds, you need fresh habitat mapping, predator management plans, and clear rules for how these animals interact with farming, tourism, and housing. Otherwise, you are dropping a fragile, expensive flock into a landscape that already beat them once. Then there is the money. Sequencing, genome assembly, lab work, and surrogate management already run into the millions across years. Add in habitat restoration, predator control, monitoring, and public engagement, and the budget climbs higher. For the same amount, conservation teams could protect and restore habitat for living prairie chicken populations in multiple states, tighten predator and disease control, and buy time for species that are sliding towards the same cliff the heath hen went over. If you take anything away from this section, let it be this. Same money, two options, thousands of live prairie chickens now or zero heath hens later. That's the real trade. Aftercare is the part that rarely makes it into headlines. A reintroduced heath hen-like flock would need monitoring for decades, genetic tracking, disease checks, and repeated habitat work as climate and land use shift. That is the slow, quiet grind that follows every glossy press release. If the funding streams run dry, you risk watching a second collapse, this time with edited proxies instead of original birds. From a distance, that might look like the project that died twice, playing out in real time. Call here, expensive reboot, fragile stage. A lot of the heath hen roadmap leans on technology that, while promising, is not yet plug and play for wild birds. Ancient DNA is one example. Museum skins and bones are harsh environments for DNA, and although modern sequencing can stitch together broken fragments, assembling a high-quality genome is still a careful, iterative process. You can get a good enough map for some questions, but the finer you push, the more gaps and uncertainties you meet, especially in regions that control complex traits like immunity or behaviour. Then there is avian germline editing itself. In mammals, cloning and embryo work now have long track records. In birds, the basic biology is more awkward. Embryos develop in hard-shelled eggs outside the body, and the pathways to reach and edit primordial germ cells are more complex. Lab teams have learned to culture and edit germ cells in chickens and quails, and a few have pushed those techniques toward other species. But we are still talking about high-skill, low-throughput procedures, not a robust platform ready for mass production of edited prairie chickens. Surrogate choice as another technical layer. The closer the surrogate's body size egg size, and incubation needs match the extinct bird, the better your odds. Heath hen work has pointed toward greater prairie chickens and related grouse as logical templates. But those birds are also conservation concerns in many places. That means you are trying to build a cutting-edge germline editing program in hosts that are not as hardy or replaceable as farm chickens. It is like trying to beta test new software on the last few laptops you own. Finally, there is the coordination problem. To move from idea to egg, you need funding, staff, labs, permits, and long-term institutional backing, all to line up and stay in sync. In the Heath Hen project, when one key lab partner was acquired and dropped out, the chain broke. Other de-extinction efforts have found new partners or built in-house labs, but each transition costs time, money, and attention. Until the tech matures into something more standardised, every project like this sits on a narrow ledge of specialised people and fragile funding. That is why, even a decade into ambitious talk, we still have drafts and plans, but no heath hen-like chicks. The science is real. The pipeline is thin. Call here, tools exist, platform missing. Now we hit the heart of the trade-off. When you build a heath hen proxy, you are almost certainly not recreating the exact bird that once boomed on Martha's Vineyard. You are designing a close cousin using genes from living prairie chickens and perhaps other species, guided by an extinct template. Some scientists openly frame that as the real goal, a functional equivalent that can fill a similar ecological niche, not a perfect copy. In a changing climate and a human-dominated landscape, that may be the best we ever do. But look at the living cousins. Greater prairie chickens and other prairie grouse are declining across much of their range, pressed by habitat loss, fragmentation, and shifting fire regimes. For them, the most useful tools are boring and proven. Protect and restore grasslands, manage grazing, control woody encroachment, and keep wind farms, roads, and housing away from key leks. None of that makes headlines like talk of resurrecting a lost bird. Yet it moves the needle for thousands of birds right now, not for a hypothetical flock 20 years from now. There is also the question of what the genetic work is for. The same genomic tools used to study heath hens can help map diversity and local adaptations in living prairie chickens. They can flag populations that carry unique traits, or that might be good donors for genetic rescue. The lost bird's DNA becomes a reference library and a warning story, while the day-to-day work focuses on keeping existing birds out of the extinction queue. So here is the sharp question. If you have limited money, staff, and public attention, where does it do the most good? Do you pour it into a long, short proxy for a bird that is already gone, or do you spend it on a habitat and genetic work for cousins that are still barely hanging on? If you take anything away, let it be the number. Millions spent, 0 chicks. That gap matters. Help the cousins first is not a romantic slogan, but from an ecosystem first view, it often gives you more song, more feathers, and more living birds for every dollar. Call here. Proxies are interesting. Saving prairie chickens is urgent. So what do we do with this project as it stands? On one side, there are clear wins. The Heath Hen case pushed genome work on prairie grouse, sparked serious debates about proxies versus true revival, and gave Martha's Vineyard a way to talk about its ecological history in concrete terms. It also helped refine avian de-extinction roadmaps, making later projects more honest about bottlenecks, welfare, and cost. Those are real receipts, not just vibes. On the other side, we have the core outcome. No embryos, no chicks, and a lab pipeline that stalled when a key partner left. The genome resources remain, the science discussions continue, but there is no active, fully funded program marching toward eggs in an incubator. Every year that passes, without that engine being rebuilt, tilts the odds further towards this remaining a case study rather than a comeback. In the meantime, prairie chickens and other grassland birds keep sliding, waiting on the kind of basic land protection that does not need any gene editing at all. From a welfare and cost point of view, the current balance is not there. The surrogates would shoulder heavy stress, the habitat is tricky and politically complex, and the long-term aftercare would demand a funding stream that does not yet exist. For a species that is already gone and that has living cousins in trouble, that is a hard package to justify as a top priority. If you put it on a funding shortlist next to live species and urgent habitats, it struggles to climb. So here is the call for this episode. The Heath Hen De-Extinction program, as currently structured and funded, does not earn a full green light. The science is valuable, but the project itself sits in a poor state with an incomplete engine. Verdict today: program paused, science incomplete. Veto for implementation now. Greenlight only for using the data to guard the cousins. The Heath Hen story matters because it shows us that extinction is not undone by enthusiasm alone. You can have community meetings, donors, draft genomes, and still end up with no birds in the brooder. That is not failure in the sense of fake science. It is failure in the sense of a mismatch between ambition, tools, and resources. If millions in plans can still leave a project at 0 embryos, that should make all of us a bit more sober when we read grand promises about bringing species back. Wild ones, that is the lens we are going to keep using in this series. Receipts first, verdict later. Some programs will edge closer to a true green light, others will stay in the not yet or not this way column. Our job is to look past the hype and check what is actually happening in the lab, in the field, and in the budgets. If that sounds like the kind of straight talk you want more of in your feed, you know what to do. On the Christmas Island rat episode, one of you summed it up perfectly. Veto, but I doubt anything will actually stop this research. And you're probably right. The lab work will keep grinding on. The verdict in this series isn't about banning curiosity. It's about where we draw the line on real animals, real habitats, and real budgets. So here is your question to take out of this episode. If you had one extra conservation dollar to spend, would you put it into a paused Heath Hen De-Extinction program or into keeping living prairie chickens off the same cliff? Let me know what you think, and then join me in the next case where we take another bold de-extinction promise and ask the same simple question. Does it really deserve to live again? Now, here is another video I know you will love. I'll see you over there, and thanks for being one of the wild ones in this series.